Good morning and happy Monday, everybody. Today we're going to take some notes about meiosis, which we spent last week starting to get an introduction into. We're going to take these notes to provide you with a one-page summary of the steps of meiosis and how meiosis happens. I think that it's a really good idea to now go ahead and pause me, go ahead and grab a piece of paper, 4 different colored writing utensils, and then once you have those supplies, come on back. So once you have your piece of paper and 4 writing utensils that are different colors, I want you to go ahead and take a piece of paper, fold it in fourths, unfold it, and then draw a nice solid line to split it up. So on the front part of our notes, we're going to do the steps of prophase, metaphase, anaphase, and telophase of meiosis 1. And then on the back, we're going to do prophase, metaphase, anaphase, telophase of meiosis 2. So go ahead and set up your page like my table is showing you and like I'm holding up my example right now. Put the names of the steps, leave a big space to draw a picture of it, and then leave some space below that picture to write a quick summary. Go ahead and pause right now, set up your page, and then press play again when you're ready to start taking your notes. So let's go ahead and start taking our notes on meiosis by both drawing and writing a summary of each step. These slides are on Classroom, so if you need to come back to them, you can. And feel free to pause the video wherever you need to make sure you have your notes down. So looking at prophase 1 of meiosis 1, you want to draw this in your top left corner where you labeled prophase 1, put a nice little sketch of it down, and then take some notes. So during prophase 1, I'll go ahead and stop my video too so y'all can see the screen a little better. So during prophase 1, the nucleus dissolves. We know that prophase, the nucleus dissolves. Once the nucleus has dissolved, the homologous pairs of chromosomes have a chance to join together. The spindle fibers also start to form. Just a reminder, even though we're not drawing it on this page, interphase, the steps of G1, S, and G2 happen before prophase 1. That's a similarity between mitosis and meiosis. Both start with interphase. So in the picture to the right, we can see the cell. The cell is shown in gray. The nucleus is gone. We can see spindle fibers starting to be made as represented by these red lines joined between the centrioles. And then homologous pairs of chromosomes are joining up together. So in this picture, we can see how pink and yellow chromosomes are next to each other and green and blue chromosomes are next to each other. Homologous pairs of chromosomes code for the same traits. So pink and yellow chromosomes might code for eye color, and then green and blue might code for the ability to make a protein. Homologous pairs have these same traits on them. After prophase 1, metaphase 1 begins. During metaphase 1, the homologous pairs of chromosomes line up next to each other in the middle of the cell. So we can see the pink and the yellow have lined up directly next to each other in the cell. We can also see that the blue and the green, which are homologous, have lined up directly next to each other. This is what occurs during metaphase 1. Also during metaphase 1, crossing over can sometimes occur. Notice how the yellow chromosome now has a little bit of red on it, and the red chromosome now has a little bit of yellow on it. This symbolizes crossing over. Crossing over is where there is an exchange of DNA between the homologous chromosomes. So in other words, they're lined up so close to each other that they trade or switch DNA between each other. So during metaphase 1, the chromosomes line up in the middle. Once they have lined up in the middle, Sometimes crossing over can happen where parts of DNA are exchanged between the homologous pair. And then we move into our next step of anaphase 1. During anaphase 1, the homologous pairs of chromosomes are separated to opposite ends of the cell by the spindle fibers. So these spindle fibers are doing the pulling to separate the entire chromosome. The entire chromosome is being moved to opposite sides of the cell. Remember that A stands for away. The chromosomes move away from each other to opposite sides. Next up, we have telophase 1 and cytokinesis. During telophase 1 and cytokinesis, nuclei are formed around the separated chromosomes, and the cell is split into two separate cells. In the picture to the right, we can see that the spindle fibers are starting to go away. We can see that each cell now has a nucleus, and that nucleus has one copy of each homologous chromosome. It either has a yellow or a pink, and then it also has either a blue or a green. It has one of each homologous chromosome. At this point in your notes, you should now have the entire front page filled in with prophase 1, metaphase 1, anaphase 1, and telophase 1. Flipping to the back of your page, we're now going to talk about meiosis 2 and the steps of meiosis 2. Meiosis 2 begins with prophase 2. The order of the steps stays the same. During prophase 2, the nucleus once again dissolves in both cells. The nucleus dissolves in both cells. The chromosomes are then left free-floating in the cytoplasm of the cell. Also during prophase 2, spindle fibers begin to form in both cells. Moving into metaphase 2. Metaphase 2, M for middle. During metaphase 2, the chromosomes line up in the middle of the cell. Notice how, in this picture, the chromosomes are lined up by themselves. During metaphase 1, the chromosomes lined up next to their homologous pair. In metaphase 2, the chromosomes are lined up without their homologous pair next to them. What this means is that the chromosome during anaphase is going to be split in half into two sister chromatids. So during anaphase 2, the chromosomes are split into their sister chromatids and are moved to opposite ends of the cell. We can see that in these pictures. I can tell that it's a chromatid because it does not have the other half of the X. So just as a reminder, I'm going to sketch this on here. This is what a chromosome looks like. This is what a chromatid looks like. I can tell it's being separated to its chromatid from its chromosome because it looks like this. The last step of this process of meiosis 2 is telophase 2 and cytokinesis. During telophase 2 and cytokinesis, the nuclei are made in each of the 4 cells. The 2 cells that existed during anaphase 2 are each split, and this results in 4 haploid cells. 4 haploid cells are produced. In humans, each of these haploid cells would have 23 chromosomes. When this process occurs in a female, it results in 4 egg cells. When this process occurs in a male, it results in 4 sperm cells. That is the end of meiosis. You should now have the back page filled in. And what you need to do to wrap up this assignment is just take a picture of your front, take a picture of your back, upload it to the assignment on Classroom. If you have any questions about this or things you're still getting confused on or you're finding challenging to really understand, please feel free to leave a comment on the assignment or rejoin the main meet and ask, and I'd be happy to help you out. Otherwise, I hope this was helpful, and I hope you have a good rest of your Monday.